You found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Hey there, YouTube, the Dapper Dinosaur here. When last we left off, Pam Acker was just showing us that she has no idea what predictions evolution would make, and for that reason, pretending that genetics didn't provide support for a common ancestry because of things like cows being more closely related to whales than horses, which, is exactly what evolution would predict, since whales and cows are both artiodactyls, unlike horses. Anyway, she's got her own explanation for the similarities and differences in organisms. Let's see if the answer amounts to magic with no explanatory power.
So I have a different explanation for similar structures, and it includes similar genes because genes code for very tiny structures called proteins, and they also have to have similar structures if they're going to have a similar function.
But do they? It's time to introduce the concept of synonymous codons. As you probably know, DNA is composed of 4 nucleotide bases, adenine, cytosine, guanine, and thymine, which pair up so that adenine is on one side of the double helix while thymine is on the other, and guanine is on one side while cytosine is on the other. RNA uses uracil instead of thymine, but that's not really relevant here. So in a gene, these nucleotides are arranged into groups of 3, called codons. Yes, I know, it sounds like codon is a neuter word from Greek whose plural should be coda, but whatever. For this discussion, I'm going to use the first letter of the nucleotide's name, so A, C, T, and G. One of the more obvious things about this whole codon thing is that there are 4 cubed possible codon, so 64. Now, there are 22 amino acids, and the need to start and stop transcription, meaning that we really only have 23 functions for a codon, since start is also just a methionine amino acid. But every possible codon has an assigned function. What this means is that of mathematical necessity, some functions are coded by multiple codons. For example, the amino acid leucine is coded for by the codons UUA, UUG, CUU, CUC, CUA, and CUG. Indeed, only the start or methionine codon and the tryptophan codon are unique. All other amino acids and the stop function are coded by more than one codon. This means that for any sequence of amino acids in a protein, there are numerous ways to code for it. For example, if I came up with a random amino acid sequence of just 10 amino acids long, we would have, say, methionine, glutamine, histidine, tyrosine, threonine, valine, glutamic acid, another histidine, glycine, and proline, then of course a stop codon. The number of codons that could be in each place is 1, 2, 2, 2, 4, 4, 2, 2, 4, 4, and 3. This means that the same sequence of amino acids could be coded for in 24,576 different ways. And from just looking at the amino acid sequence, there's no way to know which way it was coded. No natural protein is as small as only 10 amino acids, at least none that is known about. Now there are some patterns. For example, codons that start with cytosine and have cytosine in the second spot code for proline, regardless of the third nucleotide. But still, that means that nucleotide is free to vary. So what can we make of this? Well, we can differentiate between the common design and common descent hypotheses. So with common design, we know from experience that designers tend to copy whole sections of design that work well. That's why, for example, many computer programs share identical lines of code. On the other hand, we should expect that if there's a protein that was likely not shared in an ancestor species, but is shared in 2 species today, that they should have essentially maximum differences in terms of which codons code for the sequence. Now, by chance, some of them will be the same, but it shouldn't be more than we'd expect by chance. But if a group of species likely has a common ancestor that had that protein in question, then we should not only see more commonality than chance can account for, but we should also see the pattern of variation match closely with the pattern we get for a cladogram when we take other things like the rest of the genetic variation or morphology into account. And guess what? We find the prediction of common ancestry is met. But of course, this also raises the question, if 2 organisms share the same protein but have very different genes coding for them, does that mean it was separate design, meaning we can infer multiple designers? Now, I know Pam will say no, because she believes there is ultimately only one designer. But other than her prior theological commitment, why would this not indicate multiple designers? I can't think of a reason, although I'll grant perhaps there is one. If you can think of one that doesn't require a prior commitment to monotheism, let me know in the comments.
So you've probably never seen these 2 pictures before, unless you were at the Kobe retreat and you've already heard my talk. But even though you've never seen these 2 pictures before, I guarantee you know what they are.
They look like profoundly uncomfortable chairs.
Banana peel? A banana peel? No, not quite.
Well, I guess we know what Pam's guarantees are worth. Nothing.
They're chairs, right? How do you know they were chairs?
Well, I thought they were, but apparently someone else thought it was a banana peel on the left, at least. But I knew it because I know that creationists love to stupidly compare things that cannot reproduce with variation and inheritance to things that can. Because for some reason, the fact that some things are designed is an argument that anything else is designed, even though that doesn't follow at all. Based on that knowledge, I figured the only practical purpose I could see was for sitting on. Plus the one on the right has a cushion where the seat would be.
Well, they had a place to sit, a place to put your back. That one's even got a place to put your feet. It's a little weird looking. And so even though they're very strange, you can tell that they're chairs.
Honestly, if I just saw the one on the left in isolation with no sense of scale, I probably would have guessed it's an abstract sculpture before guessing it's a chair.
All chairs are going to have a certain similarity to them because they're designed for human beings to sit down comfortably without your feet dangling above the floor, ideally.
Are they? There are bar stools, rocking chairs, tree stumps, random logs, iron benches, etc. Really, the only thing that chairs have in common is that they tend to have a horizontal surface. They can have no legs, 3 legs, more legs, a back, no back, a cushion or no, and be of virtually any size or height provided the height is reachable. In some places, like East Asia, they barely even use chairs, instead simply sitting on cushions on the floor.
And support your back a little bit.
That part is extremely optional.
So things that are similar in structure not because of common ancestry. These two things didn't come from the same chair. They were designed for the same purpose.
Yes, this is why both insects and mammals have legs. But you might notice that they are not similar in their structure. For one thing, mammals have their skeletons on the inside and insects have it on the outside. But they're made of different numbers of joints, muscles, etc. Further, insect joints can only bend on one axis, whereas many mammal joints, like the hip or ankle joints, can bend in multiple directions.
Right? Wings look similar because they're made for flying.
Yes, but why are they made so differently? Clearly a bird wing and a pterosaur wing are not made the same way. So if they were designed, was it by 2 different designers?
Those single-chain antigen receptors in camels and sharks look similar because they're made to do the same thing. They're part of the immune system.
I wonder what we would find in terms of the difference between the genes that produce them. Would they have the same genetic sequence, or very different ones? I don't know, but it would be a test between common design and common descent.
Right, cytochrome C, which is very similar across all kinds of different animals, in order to live in a world where there is oxygen, you have to have that protein. In order for that protein to function, it has to have a specific kind of sequence.
Meaning it's going to be highly conserved, so evolution would predict that there's only a small space in which the protein can vary, and if that protein is vital to survival, then that protein is going to be very similar even in very distantly related organisms. To be fair, common design would predict this too, meaning that talking about such vital proteins is useless when trying to debate between common design and common ancestry. To differentiate between them, we need to focus on aspects of biology that the two ideas would make different predictions about. For example, neutral variation should fall under a nested hierarchy, if common ancestry is true, and if it is not, then it should not fall under a hierarchy at all except within the putative kinds. Spoiler though, it falls into a hierarchy, meaning that common design can be rejected.
It's not going to change very much from that, because if it did, it wouldn't function.
That's true.
So when we look at things and we look at similarities across either DNA or structures, we should be thinking in terms of what's the function here, because the function is what's driving the similarity.
Which is why we really need to look at the DNA that has no function beyond simply existing. That's where mutations simply accumulate rather than being weeded out by selection. Even if we assume that the creator put in identical non-sequence constrained sections of DNA, the mutations would only follow through within groups that the creator made in the first place, and so they should not share mutations across these groups. But we find no such groups. All organisms can be organized with neutral variation into hierarchies that are far larger than creationists will accept.
It's not common ancestry.
That doesn't follow. This fails to account for analogous structures which are not well explained by common design, but are easily explained and are predicted by common ancestry. Further, even if we only focus on homologous structures, this prediction is the same between common ancestry and common design, so it can't be used as evidence when attempting to discriminate between the two.
Okay, I have to skip this one in the interest of time.
Oh no. I won't have to explain why the Miller-Urey experiment wasn't designed to make life, nor is it exactly cutting edge in origin of life research.
I'm very sorry, but I'll go through it very quickly. So the Miller-Urey experiment was done back in the 1950s, I believe.
Some 7 decades ago. Why not talk about a more recent experiment that builds on those decades of research? Oh, right. Because that might show too much progress, and the creationists would rather that people think no progress has been made in the nearly 3/4 of a century since.
To demonstrate how life could have arisen in a warm little pond?
No, it was just to figure out if it were possible to create amino acids abiotically. It was successful in finding an abiotic means to create amino acids. It turns out that this particular technique probably doesn't have much to do with how the first amino acids actually occurred on early Earth, but there have been subsequent experiments that have found other abiotic ways to create amino acids that are more likely to have been relevant on early Earth.
So this gentleman in this picture, he was younger then when he did the first experiment, but he actually put some gases in a flask. He put a spark of lightning through them, simulate lightning, and he created amino acids. And this was hailed as like the definitive experiment that life could have come from non-living matter.
By whom? It certainly answered a big question about the origin of life, which is, are amino acids, which are essential building blocks for life as we know it, things that can be created without life? Or are they a true chicken and the egg problem with no apparent natural solution? Complaining that this didn't solve the whole problem of the origin of life is rather like complaining that golden rice didn't solve worldwide hunger. It was never supposed to, but it helped.
Because amino acids are sort of on their way to becoming life.
I've never heard anyone say that ever. What they are is a necessary condition for life. Clearly, you can't just put a bunch of amino acids in a flask and then have new life pop out. Nor is that anything that anyone has ever said, near as I can tell.
Now, we're not going to have time to go into all the problems with going from organic materials to an actual cell. That's an incredibly complicated problem that hasn't been solved by anyone, biochemist or biologist.
That's true.
But remember, an unsolved problem doesn't mean you get to just say that God did it and walk away. That there is something about life that is not currently explained just means it's unexplained, not that we have warrant to say that the explanation is a miracle.
But this problem is how to go from inorganic matter to organic matter.
Kind of? Organic matter is basically just any carbon compound that isn't carbon dioxide. It's about how to get amino acids specifically without having them built by living cells. Creationists just never managed to accurately explain the Miller-Urey experiment. It's like doing so is against their religion.
Which is an even bigger problem, it turns out, because what Miller and Urey put in their flask was water, methane, ammonia, hydrogen gas, and carbon monoxide.
Fun fact, methane is an organic compound that has many known abiotic sources. So they started with organic matter and got different organic matter, because this was never about making organic matter from inorganic matter.
And there are a couple of problems with that mixture, because what we've discovered, two important things that have been discovered in geology, looking at rocks and drilling down through rocks and examining the elemental composition of rocks, is that there would have been oxygen in the early atmosphere.
Virtually none. The real problem is that the atmosphere wasn't chemically reducing, whereas the assumed atmosphere in the experiment was. However, we know from experiment theory and observations that amino acids can form in many places, like underwater volcanoes, even in the depths of space. So like I said earlier, the exact method used in the Miller-Urey experiment is very unlikely to have been how the first amino acids formed on Earth. We have other options, though, which we know do work.
Now, most of you have probably never heard this before because it's very detrimental to evolutionary theory, because if you have oxygen in that atmosphere, if you put oxygen in that flask, you don't get amino acids, you get goop.
You get goop either way, and I don't know what most people hear about it, but if you're at all interested in topics surrounding the origin of life, you're going to hear about this. Heck, I'm not particularly interested in the topic, and I still heard about it.
That cannot have any biological function whatsoever, because the amino acids continue to oxidize, they react with the oxygen, they burn, basically, and they become non-biologically relevant.
Yeah, because they're no longer amino acids. Oxygen is slowly killing you. It's just that it's doing so way slower than you die of cellular starvation without it.
And another problem with this flask is that the atmosphere of primitive Earth probably wouldn't have contained any hydrogen, because the hydrogen would have been too light to stay in the atmosphere in sufficient concentrations. And if you don't put hydrogen in the flask, you don't get amino acids either.
It's also very reactive. And yeah, there's a reason that no one today is using the Miller-Urey experiment as if it's how amino acids formed on early Earth. That was some 70 years ago or so. We've moved on.
So you have oxygen, which means no amino acids. You don't have hydrogen, which means no amino acids. Two strikes, you're doubly out.
For this method, yes. Pointing out that you can't make a cake by mixing flour into a public pool doesn't really mean that you can't make cake at all.
So why did it work? He put the materials into his flask that he needed in order to make all of the amino acids. So you need carbon, you need hydrogen, you need nitrogen, and you need oxygen to make all amino acids except for two, which are cysteine and methionine. You also need sulfur for those. And there was no sulfur in his flask. And so it's very hard to explain, based on his experiment, how you could have gotten to a system where proteins all start with methionine, which contains sulfur, and most of them are held together by disulfide bridges between cysteines. So the very structure of proteins that we have today is not really explicable based on the Miller-Urey experiment.
Yes, it's almost like there were 70 more years of research after that. Also, wasn't she supposed to be skipping this? I feel like we've been going over it at length.
And I'm hitting buttons accidentally. I'm not sure how that's happening.
Maybe it's the forces of hell arrayed against Pam to stop her spreading the truth of creation by lying about science.
Technology gremlins or something. And just for those very nerdy science people out there, amino acids also are incorporated in your body. They're chiral. You can't impose one hand over the top of the other and have it be a perfect match. They're actually mirror images. Amino acids work the same way, and only one of those mirror images will work in your body.
Yep, and this is a well-known problem in the origin of life. It is, however, not a problem without a solution. For example, in space, polarized light can encourage the predominance of one chirality over the other, and on Earth or in space, autocatalytic cycles can encourage the formation of an amino acid of a particular chirality, while suppressing the formation of amino acids of the opposite chirality.
Not both of them. And there's never a protein that has a mixture. Proteins are all D-amino acids, not L-amino acids. Unless I said that backwards, which is possible, because if it's a true-false question, I will get it wrong. But it's all one way or the other. And there is no known non-biological mechanism to sort these out from each other.
I mean, I already mentioned two, both of which are prebiotically relevant, so, yes, there are.
So for the first protein to sort of have arisen in a warm little pond in a rock somewhere and incorporated only one kind of chiral amino acid is pretty much impossible.
You know, unless the little pond had been undergoing autocatalytic reactions for a while, resulting in a homochiral or nearly homochiral mix of amino acids. But also, the first protein probably didn't form in a pond from a bunch of amino acids. It was probably formed based on the action of RNA, which can form spontaneously and can catalyze protein synthesis on its own. Now, I don't want to give the impression that all of these things are just no big deal and that there aren't any unknowns about the origin of life. This is still an area of science where little is known with high confidence, and there are many questions yet to be answered. But the fact is that the things that Pam is saying are insurmountable problems are actually quite surmountable, and we've known this for a while now.
Okay, that was my super fast explanation of life.
Not nearly short enough.
So to sum up my earlier presentation / now presentation, evolution has failed thus far to establish that natural selection can produce anything other than variation within a kind.
That's not even a sentence that means anything, since there's no such thing as a kind, and no mechanism providing for a barrier to natural selection has been demonstrated.
So giraffes produce giraffes. They might be slightly different giraffes, but they're still giraffes.
Yes, evolution requires monophyly as a big part of how it works. So we should not be surprised to find living organisms reproducing without violating monophyly. Indeed, if a giraffe evolved into something that wasn't a giraffe, that would call evolution into question.
It's failed to provide any evolutionary sequences that aren't full of gaps.
If your elitist East Coast evolution is real, why has no one found the missing link between modern humans and ancient apes?
We did find it. It's called Homo erectus.
Then you have proven my case, sir. For no one has found the link between apes and this Homo erectus.
Yes, they have. It's called Homo habilis.
Aha! But no one has found the missing link between ape and the so-called Homo habilis.
Yes, they have. It's called Australopithecus africanus.
Oh, I've got you now. Fair enough. But where, then, is the missing link between apes and this Darwinius macilli? Answer me that, Professor.
Oh. Okay, granted that one missing link is still missing. But just because we haven't found it doesn't mean it doesn't exist.
Pshaw! Things don't exist simply because you believe in them. Thus saith the almighty creature in the sky!
Think back to our whales.
You know, just because this is just about lying that there are no transitional species, here it is.
la la
It's failed to demonstrate the antiquity of dinosaurs.
That's geology, not evolution, but also, yeah, there's no question about the antiquity of Mesozoic dinosaurs. Of course, there are modern dinosaurs too, so not all dinosaurs are from a long time ago.
Or their existence before modern animals.
All non-avian dinosaurs are extinct. All modern living animals are not extinct and currently alive. Therefore, they live after the non-avian dinosaurs went extinct. Also, you notice how she didn't name one single extant species that was alive during the Mesozoic? Not a single one. Instead, she named broad groups like octopus or lizard.
It's failed to even define what homology is in a way that isn't completely circular.
Nope, you can just use embryology to determine if there is homology. That's how it was done before evolution was widely accepted.
It's failed to build a tree of life.
No, it hasn't. It's actually not that hard a thing to build. But if your requirement is that it have 100% statistical likelihood, then sorry, that's not how science works. There will always be at least a few options for the tree of life that disagree in some details, but which are well supported. That's how life is in this complicated and impossible to perfectly measure universe.
And it's failed to replicate pre-life conditions that could give rise to biological molecules, and Captain Picard is less than pleased with his...
Okay, that's also not evolution, and yes, it is technically true that we don't know exactly what conditions led to the formation of life, and we may never know. We certainly know a lot more than what Pam has allowed for, and also, this isn't part of evolution. Evolution would work the same way if life were poofed into existence by God.
So moving on to part 2, I'm going to talk a little bit about mutations. So if you've heard anything about evolutionary theory, you've heard about how mutation for evolution, because evolution requires new genetic information. It requires something to be there that wasn't there before. You cannot get a man from an amoeba if you don't have an increase of genetic information.
Ah yes, genetic information, that oft-talked-about, never-defined term. Tell me, which has more information, a redwood tree or a blue-ringed octopus? How do you know? What units did you use? How did you measure? Of course, there is no answer that creationists will ever give. In fact, when Paul Price, a creationist PhD, was asked directly how to do this, he declined to answer.
How are you defining information? Because we need a quantitative definition of information in order to make this work.
Yeah. So, don't think that's
Emphasis
correct.
on quantitative.
Yeah, that's not correct, and that's the reason why that's the reason I co-authored the article entitled "What Would Count as New Information in Genetics." We kind of delved into this because I know that having had these conversations with you for many years, I know that this is a direction you want to go with it. Is that you want to try to ask us to quantify information.
Yes.
And,
Yes.
we don't shy away.
Let me just take a second here to be fair. I'm asking you to quantify information because your argument is that information is declining. Right?
Yes.
That's your argument, is that information is declining. So I'm saying, okay, that's your claim. If you can say it's declining, you need to be able to quantify it. So you can measure it at time point A, and at time point B, and at time point C, and tell us how we know that it's actually declining, right? So yes, it is your job as the genetic entropy proponents, right? You and Dr. Sanford and Dr. Carter and anybody else to quantify the information you say is declining.
Yeah. You cannot quantify conceptual information, and that's what we're talking about here. Okay. We're not talking about Shannon information.
There we go.
We're not.
Right. The only currently identifiable source of truly new information or new variations in the genome is mutation. So there are a couple of other ways that you can mix up information that's already there, but the only way to get new stuff is for there to actually be a change in the DNA sequence.
I mean, except for the whole thing about information, yeah, the source of new genetic variation is mutation.
And in case you haven't taken high school biology in a while,
I took college biology much more recently, in fact.
I thought I would do a quick review of DNA. So DNA, you can think of it analogously as a set of instructions to build an organism. So your DNA contains all the instructions necessary to build you with your particular eye color, your particular hair color, your particular height.
That's mostly true, but many aspects of you are actually environmentally influenced. For example, it's pretty well known that humans in developed countries are significantly taller on average than their ancestors from just a couple centuries ago. This is primarily because of greater food availability, not because of evolution. Similarly, things like bone shape, bone density, bone thickness, muscle strength and size, hair thickness, etc. can be affected by the environment. The potential phenotype of an organism is constrained by the genotype, but it's not entirely determined by it. This is called phenotypic plasticity, and you can see it if you've ever seen dogs of the same breed in different climates. In warm climates, they tend to have sparser underfur and shed more, whereas in cold climates, they tend to shed less and have thicker undercoats.
Although that's influenced by environment as well, nutrition and things like that. But you can't grow sort of past a certain size based on your genetics.
Oh, sorry. I just assumed that she'd screw that up. My bad. You know, I script these as I go, and while I do usually watch them at least once all the way through before I decide to make them into a video series, in the time it actually takes me to start writing responses and gathering resources and whatnot, I usually forget what they say. After this, she just explains what a nucleotide is, and she mostly gets it right. And I also explained that, and in more detail earlier in this video, so since I have no commentary there, including it would be iffy in terms of fair use. So I'm skipping it.
The nucleotides are arranged into sequences called genes, which we can think of as paragraphs or chapters even. In our information here, needed to build an organism is contained in the genome, and so we can think of that as the book that we would be using as an instruction manual to build you.
There's a big problem with this, though. Most base pairs are not in genes. In the human genome, for every base pair in a gene, there are 32 base pairs that are not part of any gene. They may be in intergenic regions, in pseudogenes, in promoter regions, part of repetitive elements, or just kind of hanging out in the middle of nowhere, just existing. Kind of. Really, only the developmental genes are about building a new organism, but it's not like you could survive without the other ones, so I don't know. It's not like she's wrong, more that it's a bit oversimplified.
Or to build a giraffe. I'm not obsessed with giraffes. That's the other Miss Acker.
There's nothing wrong with being enthusiastic about giraffes. They're cool animals. And while they're not my favorite or anything, I get why they might be hers. Here's the thing about nerdy stuff like that. Just embrace it. Life is short, and if being a giraffe nerd makes you happy during your time on Earth, then do it. It's not like you're going to hurt someone by being enthused about giraffes. That goes for all sorts of things. If your thing is dressing up in a Starfleet uniform or knowing all the lore of Star Wars legends, then do that.
Okay, so we're going to imagine that the instruction manual for building a giraffe is thousands and thousands and thousands and thousands of pages of genetic information.
Specifically, it's about 2.55 gigabase pairs long. The genome of the Japanese canopy plant is about 149 gigabase pairs long. So does this cute little plant have more genetic information than a giraffe? I mean, I don't know, but you'd think since Pan thinks it's a big deal how much information an organism has, and how they gain or lose it, that she'd be able to say if it has more or less information than a giraffe.
And each giraffe has to have his own instruction manual, and giraffes did not build the printing press.
That's true.
But so what?
So each giraffe must painstakingly copy his instruction manual from his parents. Now, if you've ever painstakingly copied something by hand that's thousands of pages long, you will have done something that every other human being has done. You will have made a mistake.
Yep, that's why there are so many manuscript variants in texts like biblical texts.
There's an introduction of mistakes into the instruction manual as it's copied by hand, just like there's an introduction of mistakes into the genome when it is copied by the cellular machinery that replicates DNA. So we can think of a mutation more or less as a copying error in the instructions, the codes for a living organism.
Yes, but there's an important caveat. An error assumes that there is a correct version, and while in an instance of genetic duplication, you can say that the original genome is the correct version, it's not correct in some kind of teleological sense. It's just the copy that existed earlier, and it isn't correct or incorrect, it simply exists. Mutations aren't errors in the sense of degrading some correct original, they're just changes.
So you may be wondering at this point, wait, I thought we were talking about new information, not about mistakes.
And now this is why I made sure to give you that caveat.
And you're on to something, but we'll get there. Okay, so mutation can lead to something called speciation. Now, I was actually discussing a little bit with the gentleman in the hallway about the difference between speciation and kinds.
That would require some definition for kind in the biological creationist sense that would allow us to not just positively identify members of a kind, but to exclude organisms from a kind. Something we don't have and will never have, because fundamentally, kinds are fiction.
So Mr. Owen talked about Noah taking all the different kinds of animals on the ark. He was not talking about him taking all the different species of animals on the ark. So we have seen the development and can see the development of individual species, which means that I've got two individuals that are no longer able to reproduce.
But that's just accepting basically all of evolution, because that's all macroevolution is. It's speciation. Once you accept that speciation occurs, there's no barrier to universal common ancestry provided enough time. And we know that we have plenty of time, or at least we can say that nothing that has ever been provided by a creationist has ever managed to be a problem for the scientifically determined chronology.
And you can actually even see this in the domestication of the dog. So for example, the Chihuahua and the Siberian Husky are not able to reproduce naturally.
I mean, you might get a really ambitious Chihuahua and a very accommodating Husky. But really, yeah, they're probably not reproducing anytime soon, short of artificial insemination. There's a mechanical barrier to reproduction.
There's a prezygotic isolation there, so technically these two things would be considered different species, even though we know they're both dogs.
Yeah, that's not a terribly unreasonable take if you look at them in isolation. But because both populations are in a continuum of morphology throughout which gene flow is occurring, we treat them as part of the same species, because there is in fact a path for Chihuahua and Husky genes to meet, just not through the direct mating of a Chihuahua and a Husky.
This is one of the weird ways the biology. You can see over many generations, if you have a lot of copying errors, if you have one individual drifting very far away from the mean in one direction and another individual drifting very far away from the mean in another direction, eventually these two individuals are going to be so different that they can no longer reproduce and you will have a new species.
I think she's got it. I think she's got it. By George, she's got it. By George, she's got it.
So mutations can explain that, can lead to reproductive isolation.
Well, let's all welcome Pam to accepting the main features of evolution. She's already accepted drift and selection, and now she knows that new variation arises from mutation and that speciation can occur when a lineage becomes divergent enough. That's really all there is to the basic level. Everything else is just details. It's sort of like how you don't really need to be able to do four-dimensional calculus to accept relativity.
But whether mutations can explain molecules to man evolution is another question. And
look, I get it. Pam is new to accepting evolution, so she makes mistakes. In this case, she's forgetting that it's not just mutations, it's mutations plus selection and drift, and that evolution only concerns populations of organisms, not the origin of the first life. So yes, mutation, selection, and drift can indeed explain the evolution of humans from the first life.
It seems more reasonable to me that if you're accumulating many mistakes in an instruction manual over time...
Remember, they're not mistakes because there's no one correct version.
You're not actually going to build a worse giraffe. And gradually, a worser? I don't teach English for a reason. You're going to build a worse and worse giraffe.
No, mutations are not simply harmful. Some of them are beneficial, and further, whether a mutation is harmful or beneficial is entirely based on the environment. Now, there are some exceptions. A mutation that causes an embryo to terminate before reaching sexual maturity is negative in any environment, but it's also never going to be passed down. And when speaking about this, I am largely restricting myself to mutations that can be passed down. A famous example of a mutation that is generally negative, but is positive in a particular environment, is sickle cell. In most environments, being heterozygous for sickle cell results in no particular advantage or disadvantage. But since being homozygous causes sickle cell anemia, which can be life-threatening, it is selected against, and in most human populations, the allele is quite rare. However, in areas where malaria is prevalent, sickle cell trait provides a strong resistance to the effects of the malaria parasite, allowing infected individuals to survive, recover, and reproduce. In this case, despite the fact that a full 50% of the offspring of the two individuals with sickle cell trait will either have sickle cell disease and therefore die young, or will have more or less no resistance to malaria and therefore will likely die young, the remaining 50% are more likely to survive to reproduce, so the allele is under positive selection. It's a good thing in that environment, provided that you win the coin toss, and that's all evolution needs. If some genotype results consistently in a more fit for that environment phenotype, then selection will act in proportion to the degree to which that trait increases reproductive output. That's why, according to the National Institute of Health, the countries of Nigeria, India, and the Democratic Republic of the Congo contain 90% of all people with sickle cell disease. They're all places where malaria is a big problem in some regions. So does this mean that the humans in these areas are worse? Well, the ones who die of malaria or sickle cell disease are certainly less fit in the sense of evolutionary biology, but the populations of those countries aren't in any imminent danger of collapse. So again, from the standpoint of evolutionary biology, those populations are better than they would be without the prevalence of the allele for sickle cell trait and disease. Pam's whole idea, which is just genetic entropy, falls apart with the simple observation that we just made. Mutations generally have no fixed fitness effect because they are contingent on the environment.
Until you can't build a giraffe anymore.
I'm not even sure what that means. If it simply means that the giraffe is non-viable, then those mutations cannot be passed down, and only those embryos free of such mutations will reproduce. Now, this might mean a higher rate of miscarriage or a failure of an embryo to even implant on the uterine wall, but placental mammals already have a low rate of zygotes managing to come to full term. For example, with humans, who are understandably the most well-studied placental mammal, the failure rate of fertilizations to come to full term is between 40 and 60%. You may sometimes see the rate of 75% mentioned, but it seems that this rate is too high and derived from in vitro rather than in vivo data, the latter of which are harder to obtain, hence the large margin of error. But even at the low end, 40% is a high value. I'd not like to know that I had only a 60% chance of surviving the next month or so, which is what at the best a human zygote is facing. So is Pam saying that the higher loss rate of zygotes would doom giraffes? Perhaps it would. But remember, any mutation that would result in a high enough loss of zygotes to harm the reproductive output of a giraffe carrying the mutation would be selected against, and so would be very unlikely to spread in the population. Further, this whole idea of genetic entropy depends on the idea that mutations are net negative, which is to say that genetic diversity is a net negative, and the ideal for a species would be minimal diversity. But this flies in the face of everything we know about conservation, where genetic diversity is a huge priority, and animals with low genetic diversity, like cheetahs, remain at great risk even when their census population size is reasonably large.
And this is a concept that was coined by Dr. John Sanford called genetic entropy.
Called it.
And Dr. John Sanford was the inventor of the gene gun. He teaches at Cornell University, or at least taught at Cornell University.
Yeah, he's also a demonstrable liar, whose idea fails to match experimental reality and only works in a computer program he made specifically to demonstrate the plausibility of his idea. Basically, it's like if I paid someone to make a simulation of YouTube where I was the most popular channel and then tried to use that as evidence that I'm bigger than T-Series. For more on Sanford, watch my series Sal's Pal, because of course Sanford is a friend of Sal Cordova, who despite saying he'd watched the series, as far as I know, hasn't, despite pretending to respond to me, which he failed at, since he didn't know what to respond to anyway. I also did a live stream with population geneticist Dr. Zach Hancock, which you also may enjoy, in which we directly responded to Sal's attempt at a response, except, we actually watched Sal's video, a courtesy he didn't bother to extend to me.
I'm not sure if he's still teaching.
He's basically retired at this point.
He's extremely well respected in plant genetics and in the world of biology.
Maybe at one point, but then he fell off the deep end and started making up stuff that is mathematically impossible. It's rare that I ever say that something in science is proven, but Sanford's ideas are mathematically impossible and therefore have been 100% disproven. They are completely incoherent. There is no possible world in which they are true. It doesn't work. There is as much going for genetic entropy as there is for the possibility that the square root of 2 is a rational number, or that Euclidean triangles have internal angles that do not sum to 180 degrees. His ideas carry less weight by far than the flat Earth once they're analyzed.
And it wrote a whole book on this concept, this idea that mutations over time did not lead to the accumulation of new information.
Funny how he wrote a book instead of, submitting the idea to peer review. Of course, we know why he didn't. His ideas contradict not just experiment and observation, but logic itself. So there's no way anyone who knows enough to peer review a paper on evolutionary biology would approve such shoddy work. Books should come after your ideas have passed peer review, not before.
But they lead to the degradation of old information. If genetic entropy is going to be true.
Which it can't be.
It can only be truly a problem if all the genetic information in the organism is useful.
Well, since I don't know what genetic information is or how to measure it, and even Paul Price can't seem to manage to figure out if it goes up or down intuitively, that wasn't a real thought that Pam just had. How do we even know that there's any genetic information to be useful since we can neither define nor measure it? Also, I want to point out that while Sanford does talk about information in the stupid way creationists usually do, his arguments don't actually need the concept and can be made focusing entirely on the fitness effects of alleles. Of course, the problem is that once you do that, you either have to redefine fitness to just be stuff I personally like, or the idea falls apart. Creationists tend to opt to redefine fitness, at which point the proper response is, cool, well, since that has nothing to do with evolutionary biology, I don't care. Go have fun eating paste in the corner.
So I have to assume that the whole instruction manual actually codes for something important.
Wait, did we just hear Pam say that if genetic entropy is true, DNA is all functional?
Non sequitur. Your facts are uncoordinated.
If there are pages in the instruction manual that aren't important, they can accumulate errors, and that won't affect the final outcome of building the giraffe.
Yes, and that's the case. There's this thing called a knockout experiment, where whole sections of DNA are simply snipped out of the genome of an organism, and it is then allowed to reproduce. And while to my knowledge, this hasn't been done with giraffes, since they're big and reproduce relatively slowly, and are also endangered, it has been done with mice, and mice can have the majority of their genome simply deleted and go on to live healthy lives and make healthy mouse babies. I see no reason to think that the giraffe is some exception to this, although I doubt we'll ever see a giraffe knockout experiment.
Does that make sense so far?
No.
Okay, because sometimes I get lost in the weeds in this particular presentation because, it's my thing.
Her thing is using already mathematically incoherent ideas to make non sequiturs about the predicted level of genomic functions, which we already know to be false? Weird thing.
All right. So, for mutations to be the mechanism of evolution,
which they're not, they're a mechanism for evolution.
4 things have to be true. Right? And the first one goes along with that idea that's the opposite of genetic entropy. So for genetic entropy to be true, all the information has to be useful.
No, what needs to be true is for selection to be weak and for the fitness effects of mutation to be fixed. Neither is true. What the fractional function is doesn't really matter, because even if the non-functional parts of the genome accumulate mutations, so too will the functional parts. And if selection is too weak to operate on them, and the mutations are on net negative in all environments, then the species will decline in fitness as genetic diversity increases. Of course, selection isn't that weak, fitness is contingent, and fitness in the population as a whole tends to increase with genetic diversity.
For evolution to be true, most of the information has to actually not be useful.
Also not true. While it was proposed that a low functional fraction was necessary for evolution to provide a buffer from harmful mutations, it turns out that there is no upper limit to the fractional function of the genome. Now, in organisms with well-known genomes, it turns out that the functional fraction of the genome is relatively low, but evolution doesn't need it to be. It's just that there are a lot of processes in life which expand non-functional regions and convert functional regions into non-functional regions. So we predict that most organisms should have function for only a minority of their genome.
So it was initially thought that about 98% of your genome was useless, and so it's called junk DNA.
I can't speak to past estimates which are no longer current, but the simple fact that about 3% of the genome is part of genes means that it's simply impossible for 98% of it to be non-functional. About 75% is probably non-functional, probably a bit more. And this is not to say that that part of the genome has no known function, and therefore might be functional, but we don't know. I mean, we know it to be non-functional. It is not important to human life and can suffer any mutation, including deletion, without any measurable impact on the human that experiences such mutations. I wonder how long Pam is going to go before she mentions the 2012 ENCODE paper without ever bothering with the 2014 partial retraction in which ENCODE admitted that their initial definition for function was dumb.
Because only about 2% of your genome is genes.
It's more like 3%, but the genome isn't really made of genes and then junk. There are things like regulatory regions for genes, and centromeres and telomeres, which help with chromosomal folding and protection, etc.
The rest of it is not genes. And so we thought, well, all this stuff is not genes, it's not really doing anything, so it's junk.
Did we think that, or is it just Pam's straw man?
And then most of the changes that happen, most of the mutations that happen need to be harmless. They need to be neutral. They need to affect the instruction manual basically not at all.
Most of them are, but that doesn't have to be the case. The degree to which a population can withstand a high rate of mutation depends at least in part on that population's reproductive capacity. An example is the mutator lines in the Lenski long-term E. coli experiment. Some lineages of the bacteria in that experiment have extremely high mutation rates, and bacteria are known to have much higher functional fractions of their genomes than eukaryotes. This means that any given mutation in a bacterium is much more likely to have a measurable impact on fitness than in a eukaryote. Despite this, they reproduce so much that some of the bacteria by chance will hit upon extremely fit phenotypes, and then go on to outcompete the rest of the population to a large extent. Now, humans probably can't directly replicate this strategy since it takes an absurd amount of time for them to reproduce at all, and most humans, especially human females, will have trouble getting past a dozen or so offspring, never mind the hundreds that this strategy would require. But so what? If the claim is that organisms cannot have a high functional fraction of their genome or evolution doesn't work, this directly refutes that claim.
some of the changes should be good. So I can't get a new organism if I don't have new information. I can't get a change that drives evolutionary biology forward unless I have some beneficial mutations.
Good thing those are trivially easy to find.
And then the last thing is that as I accumulate changes, I'm going to have to lead to a new kind of animal, not just a new species of animal.
Well, since there's no such thing as a kind, this isn't a problem at all.
So not a new giraffe from an old giraffe, but a new okapi from an old giraffe, or something like that.
I love it when creationists demand as evidence of evolution things that the theory of evolution says are impossible. It just demonstrates the degree to which they either have no clue what they're talking about, or are just dishonestly creating a straw man. Given that Pam here has a graduate degree in a biological field, I'm leaning towards the latter.
Okay, very, very different kind of change. So 4 things have to be true. Has to be useless. Most of the information has to be useless. Most of the mutations have to be harmless. Some of them have to be good, and the accumulation has to lead to new kinds.
Well, that was mostly nonsense.
Hold on to that.
I'd rather not.
Because it's about to get harder.
That's what she said.
All right, so the first problem is that we're running out of useless DNA. So ENCODE, which is the Encyclopedia of DNA Elements,
turns out I didn't have to wait too long,
is a consortium of scientists working around the world who are analyzing the human genome sequence that was published in the early 2000s. And what they had, or the information they had accumulated by the year 2012 indicated that at least 80% of your genome is functional.
Yeah, because in 2012 their definition of function was that any activity at all was function. So according to ENCODE, if you have a knick-knack in your basement that you forgot even exists, but it accumulates dust, then the function of that knick-knack is to accumulate dust, even though you never think about it, wouldn't miss it if it were gone, and don't even really know it's there, but it's still functional apparently. I think we can all see why that's dumb. In fact, 2 years later, ENCODE saw why that was dumb and retracted that statement. Instead, they went to a more reasonable definition of function, which is that if the DNA was not sequence constrained, it wasn't functional, since if it did anything important, then its sequence would be important. And if it could undergo any mutation at all, including deletion, without an effect on the organism, then it's probably not functional. Under that definition, the genome is more like 80% non-functional, a near complete reversal of the 2012 paper. How much you want to bet that Pam ignores the fact that ENCODE wouldn't stand by her use of their work, because they don't even stand by their own use of that work from 2012?
So that means that we've gone from 98% junk to possibly 20% junk, but probably not, because I suspect that they're still finding uses for these things. And they've done it basically based on sequence comparison, and they're looking at things that are useful.
No, they were looking for any biochemical activity at all. And in fact, their estimate went the opposite way Pam thinks it should, and had already done so before she even made this prediction.
And comparing those sequences to things in your genome that are not genes, and they're saying, oh hey, that looks like a regulatory element, and that looks like it's a promoter, that looks like it's an enhancer, that looks like it's doing this thing over here, it's involved in microRNA regulation.
No, because we know that doesn't work. Just the example of pseudogenes demonstrates that. In sequence, a pseudogene looks a lot like a gene, but it isn't one because it's broken for some reason. Famously, humans have a pseudogene called the gulo pseudogene, which is homologous to the gene that in most mammals allows them to synthesize vitamin C. Humans are susceptible to scurvy specifically because this would-be gene is broken. Other parts of the genome that are not genes but are still functional can also break, so simply observing that the sequence is like that of a functional region isn't evidence of function. That's why, at first, the ENCODE project needed evidence of biochemical activity to declare function, and then later they needed evidence of sequence constraint. At no point was simple resemblance to a known functional element sufficient to conclude that a region of the genome was functional.
So all of these things, they're looking at things that they know happen in the cell, and they're saying, okay, this DNA matches DNA that does that thing. So it probably has a function, because if it has a similar structure, it has
a chance to be functional, but that's it.
A similar function, right? That's what they're basing this idea on.
No, they're not, because that demonstrably does not work. That's not a reliable way to detect function, even under the stupid first definition that ENCODE used.
So this is not, the idea I presented to you with the chairs is not a novel idea. Every biologist worth their salt knows this, but they don't apply it to evolution, generally speaking.
No, not every scientist knows that a useless test that wasn't used to detect function was used to detect function in 2012. And they do know that the actual 2012 claim was retracted in 2014. Instead, they know that somewhere around 80% of the genome is known to be non-functional, as backed up by knockout tests in mice, which as UR-clontigliers are reasonably closely related to humans.
Okay, so a couple of specific examples, because I like specific examples. I was working in the Corsi lab at the Catholic University of America, and we were looking at a model organism called C. elegans. It's a tiny worm. It's about a millimeter long. It's transparent, has about 1,000 cells, and they've tracked the development of every single one of those cells from the time it is a single-celled organism to the time it's an adult. It's a pretty fascinating thing to study.
It is, and in one of the only times you'll hear me endorse Richard Dawkins, in his book The Greatest Show on Earth, there is a very interesting discussion of the embryonic development of this worm. Every single cell in the body has a particular name. Now, it's not a name like Jeff or something. It's an alphanumeric designation, but still, it's mind-blowing to think that we have an organism that's so well studied that every single cell in its body has a designation.
And what we found was that in DNA sequences, so in genes are funny things, because in your DNA, there are little interruptions in them. They're called introns.
Yep, they have to be removed by enzymes called spliceosomes, which remove them so that only the parts of the genome that are actually coding for amino acids are read by your ribosomes.
And once the DNA gets copied into RNA, those interruptions get kind of spliced out. And so these were a candidate for junk DNA when they were first discovered, because scientists were like, well, they're not in the final gene product. They're only in the original code, so they must not be important. But we found, when we were working with C. elegans, a regulatory site in one of those introns that was important in turning a certain gene on and off. And if that gene did not get turned on, that C. elegans could not defecate and it could not lay eggs.
Yeah, but most introns show signs of not being sequence constrained, meaning that any mutation in them is meaningless to the organism. So despite the fact that some introns have function, that is not evidence that the ones that we know don't have functions do, nor is it any indication that introns have function normally. Biology is complex and messy, and there are a few rules that are universally true, but finding a small exception to a general trend in biology doesn't negate the trend.
And this is a problem because C. elegans produce eggs even if they can't lay them, so eventually the worms would die from their offspring exploding out of their insides.
Where's Warrant Officer Ellen Ripley when you need her? Also, fun fact, the character Lambert, played by Veronica Cartwright in the film Alien, is trans, which is revealed in her personnel file in the sequel Aliens, which says that she was born male but underwent a gender reassignment procedure. Xenomorphs don't discriminate on the basis of gender identity. They're equally murderous to everyone.
Which is a horrible way to go.
Yeah, Kane seemed to not enjoy it.
So this is clearly an important region in the DNA, because if there's a mutation in this one spot, in this intron that was thought to be junk, that causes this worm to die this horrible, terrible death.
And did she find similar things for all the other introns, or did she just find one important intron and then just inappropriately extend that to the whole of introns?
And to be very uncomfortable in the meantime.
Lifelong constipation and pregnancy is like that.
And that's what that slide was about, actually. And I'm going to skip that one, too, because it's super technical.
Really? She couldn't have gone through alternative splicing and instead skipped over the Miller-Urey nonsense? I do not like these choices. But you know what? Let's come back later when I'm better mentally prepared for all of this. If you liked the video, hit like and tell me in the comments. If you didn't, feel free to hit dislike and tell me what the problem was in the comments. Either way, please remember to subscribe and hit that bell icon so you're always notified when I have more content. I'm the Dapper Dinosaur. Hey, before you leave, I just want to take a second to thank my patrons and channel members, especially those pledging $20 or above. Ben Tovend, Phil Ravara, Tapioca Weasel, Whispers, Denny5252, Eleron Teller, Horseflesh, Ian Chen, Kelvin Brostock Van Manen, Landon Knoll, Mabbity Babbity, Monkey They Them, Robert Carpenter, Sphincter of Doom, Vollus, Strawberry Vein, and Ita V. It's because of my channel members and patrons whom you're seeing on screen that this channel can stay afloat. Without you, it would all shut down. If you want to join the team, there's a link to join the channel below this video, and there's a link to join the Patreon in the description. On the Patreon, you can get a 10% discount for pledging annually, and either way, you get early access to virtually all of my scripted videos, often 3 to 5 months before they come out for the general public. Thanks for watching.
How